talking about spore formation. Spores are usually produced in sporangia. Sporangia is a body or a structure which, if in singular, we call it sporangium. Now, these are non-flagellated spores. Non-flagellated, non-motile means cannot move on its own, which cannot swim also, and these spores are basically known as aplanospores. And if they are on the other hand motile and flagellated, they have flagella and they are motile, they are known as zoospores. These are the two categories. Clear? So basically, what is a spore? It's a single reproductive structure that detaches from the parent and give rise to a complete individual. And reproduction by spore formation is a common method of asexual reproduction in some bacteria and most fungi. Clear? What happens during the formation? See, whenever the formation of spores takes place, whether it is a planospore or a zoospore, the fungal hyphae develops a large sporangium. This is the fungal hyphae, which is on a substratum. Substratum, we can say substratum can be anything. It can be soil, it can be a bread mold, it can be any rotten fruit. So we call it substratum. That means the solid support onto which this fungal hypha has to grow. It develops an enlarged sporangium here. Clear? It may not be one, there can be 2 or 3 sporangia simultaneously growing on the same hypha. And this nodulated structure will not end up here. It may grow further, and wherever favorable conditions are, it may give rise to another blob-like structure known as sporangium. And on the lower side, it will develop root-like structures known as rhizoidal hyphae. This is going up, so we call it stoloniferous hypha, and the one which is growing downward, this one, we call it rhizoidal hypha. Alright, so we mark it stolon. And these are rhizoidal hyphae because these are towards the root side. Then this ball-like structure is known as sporangium. And this entire stalk and a sporangium together known as sporangiophore. Now what happens? The fungal hypha develops a sporangium, and the nucleus of the developed sporangium, it divides several times. And each nucleus gets surrounded by a bit of cytoplasm, as we have done in multiple fission. It is surrounded by a bit of cytoplasm and develops into a spore. These spores mature inside the sporangium only, and they are liberated by rupturing of the sporangium wall and dispersed to grow on a new substratum. Each one, how do they first of all move out? This is how when the mature sporangium, it receives favorable conditions. All these spores, these fall on the substratum, and each one grows into a new hypha and germinate in this new habitat to bear a sporangium. Again the same process starts. The examples for this kind of spore formation that we are discussing here are Rhizopus, fungal species, Mucor, another fungal, Rhizopus mucor aspergillus, and Penicillium, Penicillium. Alright, so these are the few examples. This is spore formation in fungi and in bacteria. Now moving to vegetative propagation in plants. Vegetative propagation is asexual reproduction in plants, in which the most important thing that we have to keep in mind is paths other than seeds are used as vegetative propagules. That we have done, like the structure unit that is employed in place of a seed. It may be a stem, it may be a root, it may be a bud, and it is a method of propagation in which plants have lost their capacity to produce seeds or produce non-viable seeds. That means seeds are produced but they do not have the capacity to germinate. So it is very essential for such plants. Example, banana has non-viable seeds. It's not a seedless plant. Then seedless grapes. Third we say rose. And then is pineapple. Here seeds are present, but we cultivate vegetatively just for its better maturity period. Then we talk about that among the flowering plants, every part of the body like root, stem, leaf or bud, all these can take part in vegetative propagation and all are the vegetative propagules. And there is a natural method of vegetative propagation which is going on, and this method is number one is tuberous roots that we know. Example, sweet potato. They naturally grow in this method. Then in guava plant, underground modified stem. First we will do the underground modified stem part, and that is taking place in ginger, Ecornia. It is water hyacinth. It is also known as terror of Bengal because it was badly spread there. Terror of Bengal, Ecornia crispus. Then is corms, bulbs, garlic, onion. All these are vegetative propagules which grow naturally. Then we talk about roots or the buds, leafy shoots of guava. Root buds, that can easily grow into leafy shoots in guava. And most important is leaf of bryophyllum. It grows naturally. What happens? This leaf develops notches and each notch bears a bud. You know what happens whenever this bud, it fully grows, it falls on the ground and develop into a new plant. So we say plants like in the leaf of bryophyllum, these are adventitious buds. These grow in the margins of the leaves and develop into new plant when it falls onto the ground. Generally the whole leaf falls onto the ground because of the weight of all these buds which are growing at the margins. And as soon as this fully mature, this bud region along with the leaf falls onto the ground, it develops into a new plant. Then moving to the artificial methods of vegetative propagation. In artificial methods, first of all, these are man-made, we have to keep in mind, and developed by plant growers and horticulturists for commercial propagation. Alright, so first one we talk about is cutting. Second we talk about is layering. And third is grafting. What happens in cutting? I'm taking cutting first of all. In cutting, a small part of any plant organ, maybe the stem, root or leaf, can be used for vegetative propagation. Alright, and here stems are mainly cut and employed for this propagation. Why? Because these are non-green, green, woody. When this is the section which we choose, this is the main stalk region, that means which will be bearing the green and non-woody tendril onto it or a branch onto it, which will be selected for cutting. So here the main stalk region is, or the main part of the plant is always woody and non-green, and the branch that we use for cultivation is always green and non-woody. This region of the stem is always cultivated. Okay, here when we select the plant part, we have to select for a non-woody or a green branch, which is normally collected, and about 20 to 30 centimeter long piece of stem are planted in a natural position in the wet soil in this way. It is first obtained by giving this slant cut, and then this piece is recovered and is grown or planted in wet soil. It is about 20 to 30 centimeter long piece. It is planted always in upright position or natural position, and then what happens, it gives off roots from the lower end, which one is planted in the ground. And shoot buds from the aerial nodes, that means the nodes which will come on the top. If we have selected this piece and we have grown this slant position here in ground in this manner, then what are we exposing? Since it is cleared off from the vegetation from this area, this part will give rise to rootlet area or the roots. New roots will arise from the lower end and shoot buds from the upper end. And this will not happen in a minute's time or a week's time. It will take little time to grow, maybe a month or more than a month. A cutting develops a new plant similar to the parent plant. What happens basically is why the root area growing from the lower notch only, because these cells, the apical cells which are exposed here, they are in the soil, and over there they got differentiated to form the root region, and here when the cells are exposed in this environment, they are exposed to form the shoot buds from the aerial nodes. So the shoot buds arise from this area and the root buds or the roots arise from the lower area. And rose, grapes, sugarcane, banana can be grown by the means of cutting. Now moving to layering. Layering also, where artificially stem is or the branch is induced. Here we select a plant and a branch, which one is again non-woody, green, tender, flexible. Why? So that we can pull it and we can fix it in the ground. First of all, when we select this branch, we clear off the vegetation of this area, and little vegetation is always supported on the top. Then the second step is we pull this branch and fix it in the level of ground with the help of a clamp. This is how we have fixed it in the ground. And these are the natural roots of the main plant. This is the stem, the main stem of a plant, and this is how we have clamped. With the help of clamp, we have fixed this particular one in the ground. We know that we have cleared vegetation from this area. So what will happen? New roots will arise. As when the cells will undergo differentiation, these cells are in the ground, and the area or the cells which are going to develop here will automatically give rise to the root system. And this is the part of the plant which further starts growing and shows growth. In this manner, the new plantlet after a period of time will be recovered by giving a cut near the clamp area, as the new roots are already present. So in this way, a new plant is recovered from the layering procedure. This is done when we say that roots are artificially induced on the stem branch before they are detached from the parent plant for propagation. Example, jasmine plant, normally obtained by layering, right? Moving to the third, grafting. Graft means a patch. In this grafting technique, two parts of a plant are joined, and they are joined in such a manner that they grow as one plant. So it is done between the two closely related dicot plants having the same vascular cambia. Alright, the plant where basically the rooted plant where the grafting is performed, we call it stalk. Now this is the rooted plant where the grafting is to be performed. We are going to call it stalk. Alright, so this is our stalk. And the portion of the other plant, the bud or the branch that is grafted onto the stalk is called scion. If I am going to take this plant, I'll show it by two means. First of all, this one onto which I am going to place the another part of the plant. Now I want this area to be fixed onto it. Clear? So the upper one, this one is known as scion. S-C-I-O-N. Stalk. Always remember stalk means reserve. So that is stalk, and the scion is the one, the portion that is grafted onto it. So during grafting, about a long scion is placed onto the cut end of the stalk and tied in such a way that the cambium of the two comes in contact with each other. And the joint is covered. First of all, this is the cambium region and the cambium region. It has to come in contact with each other. That's why we have given a slant cut and we have to make sure of this thing. Then the first layer, first of all, when they come in contact with each other, the joint has to be covered with the layer of wax or clay. To prevent the evaporation of water. To prevent evaporation of water. Then everything, whatever extra buds and all these supporting substances, branches are there, these need to be removed. Then after this is tied in such a manner that it has a wrapping sheet onto it, or a talc sheet onto it, and tied with a thread also on the top. Number 1, wax or clay. Number 2, talc sheet or any sheet. And number 3, we have to tie it with a thread, so as to fix it completely, so that no more material from this cambium region should ooze out through this cut. Clear? Now, what are the conditions? What are we looking forward for in a stalk? Stalk actually belongs to a wild variety. Why a wild variety is always chosen? Because we never care for all the wild plants as we do for the ornamental ones. So they have an extensive root system so that absorption of water and minerals can take place easily. This wild variety, it possesses an efficient root system. A very young scion can be made to flower if grafted on a mature stalk. Similarly, a single stalk can support many scions belonging to different varieties. Like if I choose this stalk and onto this stalk I would like to propagate many young scions. So what do I have to do is I have to keep a check on the middle cambium region, and I can graft many scions of various varieties. But each one should reach the cambium region and should join in a manner that no leakage, no oozing and no transfer of material should come out. So I can propagate different varieties and I can support many scions belonging to different varieties on the same stalk. Example, a mature stalk of citrus. Stalk we are choosing citrus. It can support the grafting of sweet orange, lemon, lime or grapefruit easily. Same way, apple, pear, peach and apricot trees are also propagated by grafting. What is the advantage of this grafting? Number one, what it helps in? It helps in combining the desirable traits. So what do we do is all the desirable traits are combined, right? Then it enables this young kind to grow quickly to produce flowers and fruits when grafted to a stalk of a mature plant. And it can be used to produce varieties of seedless fruits. Alright, there are many advantages of vegetative propagation that we have done. Quick method, new plants can be produced easily, can be grown from one plant by vegetative propagation, seedless plants can be grown. Root, stem and leaves, buds can produce and give rise to new plants whenever the mature plant cannot produce seeds or cannot produce non-viable seeds. Or what could be the disadvantage that we think? Disadvantage that mainly can be observed in vegetative propagation is it is only used in plants which cannot produce seeds or or producing the non-viable seeds. And no variation. Did you see? Only the sky region where we have seen variation. Otherwise there is no variation. The new plant exactly looks like the parent plant. Moving to the spore formation process, that means micropropagation, the tissue culture technique. Tissue culture technique. What is this tissue culture technique? Now this method includes propagation of plants by culturing the cells. We have to culture the cells. We call it micropropagation as well. In this plant, the plant cells or tissues are cultured or sterilized. That means free from microorganisms containing necessary nutrients and hormones in artificial medium. Now proper medium is important and we call this process as tissue culture technique or micropropagation. First of all, we recover the mass of cells or callus from the apical region. Callus means mass of undifferentiated cells. From the apical region of a plant. Why? Because it is a fast growing cellular mass. It is devoid of any kind of differentiation. It does not know what it has to grow into on maturity. So we recover this and transfer to a medium in a petri dish. So this is that mass which we have obtained and is transferred into a medium. This medium is always agar-agar, a gelatinous substance which will support the growth of this callus. Callus will quickly grow and from here it is transferred to another medium. In this medium, we will add hormones also. Hormones and nutrients required for the growth of the plant. So this callus got furthermore differentiated and is shifted here. And ultimately here the plantlets are then growing and give rise to a particular shape and got differentiated further. So in this it will definitely acquire a shape. These plantlets will acquire a shape and the mass or the cells will get differentiated. From here, the plantlets are recovered and are transplanted to separate pots, nursery beds, and allowed to grow for a definite period of time, meaning we have to give it little time to grow. Now then the mature ones, they will grow for a definite period and will become mature. These mature ones are recovered and are transplanted in the field. Clear? So this will be the C and this will be D. This is how the process goes from micropropagation. These, when transplanted into the field, they employed the technique for quick multiplication. This is a commercial propagation, and orchids, dahlia, chrysanthemum, carnations, these are propagated in this manner. So this is vegetative propagation in plants. Next we move to sexual reproduction.